Okay, let's do a quick review of the important concepts in the cardiovascular system that we've talked about so far. And of course, the first thing we want to always make sure we understand is that our heart has basically two different circuits that is around it. One circuit is the pulmonary circuit, and this is the blood vessels that are pumping blood to and from the lungs. Then there's the systemic circuit. This is normally the circuit we think most about because this is the other 10 systems in our body. So when our heart pumps blood to and from the rest of our body, then that's the systemic circuit. So pulmonary circuit, to and from the lungs; systemic circuit, to and from the rest of the body. Arteries always take blood away from the heart. Veins always take blood to the heart. Okay, so in a systemic vein, it's taking blood to the heart because the blood is oxygen poor. In a systemic artery, then we're taking blood away from the heart, particularly here in the aorta, and that blood is oxygenated. In pulmonary circuit, we're taking blood away from the heart towards the lungs because it's oxygen poor. And in the pulmonary veins, we're taking blood from the lungs back to the heart, and that's oxygen rich. So even though we have arteries and veins in both, whether the blood in it is oxygen poor, which is denoted by the blue, or oxygen rich, which is denoted by the red, that's different. So we do have to understand that we're talking about the systemic circuit or the pulmonary circuit. Okay, the next thing we looked at was the intrinsic conducting system. Here we see we have the SA node, the sinoatrial node. We have the internodal pathways between the SA node and the AV node, the atrioventricular node. Of course, the SA node is actually in the wall of the heart near the opening of the superior vena cava. The AV node is also in the right atrial area in the septum there. And then the AV bundle is really the connection between the upper portion of the heart and the ventricle, the lower portion of the heart. So even in this picture, even though it looks like the AV node there is actually in the ventricles, it's actually right before the ventricles. And we actually have a groove here that separates the top part of the heart. That's the coronary sulcus, which has a fibrous skeleton that keeps that signal that's moving through this intrinsic conducting system from just jumping. Okay, now we have the AV, again, the AV bundle, which is the actual connection that the signal can move from the upper portion of the heart where the atria are to the lower portion of the heart, and it moves through the septum. So embedded in the septum is the AV bundle running into and branching into the bundle branches, which have these offshoots that actually go to the papillary muscles here to cause them to tighten. Now, the bundle branches branch into the Purkinje fibers, which then runs up the walls of the floors of the ventricles and then sends the signal across, of course, to cause the contraction of the muscles. Now, remember what's going on in this intrinsic conducting system are electrical events, and it's spreading from cell to cell to cell to cell, an electrical event, but it's also spreading across into the actual muscle cells, the cardiac muscle cells called the myocytes. And of course, what it triggers them to do is to go through a contractile event. Okay, now we have a signal that goes along with this movement of the impulse, the cardiac action potential from cell to cell to cell. And in the autorhythmic cells that make up the nodal system and the intrinsic conducting system, we have this pattern. We always start out at a resting membrane potential, and then we have this area here that we can call prepotential, and we can also call it the pacemaker potential. Now notice -60, where we're starting, is going to be our resting membrane potential. And this is a gradual buildup because due to the sodium leak channels allowing sodium to leak in, and since sodium's a positive ion, then it actually starts building up the voltage from a more negative to a less negative number. And -40 would be what we would call threshold, because once sodium leaks into threshold, then that opens up fast calcium channel and calcium rushes in. And we're going to call this our depolarization here because calcium is rushing in, and we're going to pass zero. So then we're going to go into a positive voltage that we can measure. And when we cross zero, then that actually triggers the potassium channels to open and the calcium channels start shutting. So when the potassium channels open, potassium actually starts leaving the cell and creates this portion of the graph, which is our repolarization, until we fall back down to resting membrane potential, and then we repeat this pre-potential or spontaneous depolarization. Now this is primarily working and our heart rate is being established in the SA node, so it's called our natural pacemaker. And here's our SA node, our pacemaker, where our signal is initiated, and that's where the signal begins. That's where that spontaneous depolarization starts taking place. So let's look at the timing involved in this particular situation. So we start out with, at resting memory potential, and we start out at 0 time, and by the time that we start generating the signal from the SA node to the AV node, then that is actually going to take 20 or 50 milliseconds. 50 milliseconds, so 50 thousandths of a second for the signal to spread from here to those internodal pathways to the AV node. Now, there's a really important delay that happens at that AV node before we can send the signal to the AV bundle. And notice here in this picture we can see the AV bundle. This region here is what connects the upper portion of the heart to the lower portion of the heart before we branch it to the bundle branches. So again, in our countdown, it takes 50 milliseconds, very, very fast, but 50 milliseconds for us to send the signal from here to here, and then we delay the signal. And notice I only took 50 milliseconds to send the signal from here to here, but we delay it 100 milliseconds. Now the reason why it's really important that you get that delay is because remember, this electrical signal is then going to trigger the mechanical signal, which is the contraction of the muscles. And what the contraction of the muscles is going to do, it's literally going to squeeze the blood that's in our atria down to our ventricles. So if I don't have that 100 millisecond delay, that means that by the time the signal also runs down through the AV bundle branches, Purkinje fibers, and then the signal spreads across to the muscle cells in the ventricles, that basically I would be trying to squeeze blood down and blood out at the same time, and that'd be a terrible situation because I need my valves open for blood to go through it. But when I start squeezing blood from the lower area, it literally closes my valves. So I need a delay here so I have time to squeeze the blood from the atria to the ventricles before the signal moves and I start squeezing blood from the ventricles out through the pulmonary trunk and the aorta. Now again, notice my time. We're talking milliseconds or thousandths of a second. So it took 50 milliseconds to travel from here to here. Now it's a 100 millisecond delay. So that's a total of 150 milliseconds just to get the signal there and delay it before the signal can be sent through the septum into the bundle branches. Now notice it only takes 25 milliseconds for the signal to pass into the septum and across the moderator band. Again, that moderator band takes the signal to a papillary muscle, and we need to brace that papillary muscle to tighten up the chordae tendineae that's attached to my cusps of my valves. All right, but looking at our timing again, it took 50 milliseconds to get here, 100 milliseconds to delay, that's 150 milliseconds, then 25 more milliseconds. So our total time is 175 more milliseconds. Okay, now it takes 50 more milliseconds for the signal then to pass up the Purkinje fibers and across to the actual muscles here to trigger their contraction. So from the time the signal was initiated here, spread through the internodal pathways to the AV node, our 100 millisecond delay, then our movement into our AV bundle, down our bundle branches, up our Purkinje fibers, the whole time took 225 milliseconds out of a second. So that means that the rest of the time in the second before we start again will be everything going through a repolarization and relaxation phase. Here we see our EKG or ECG wave tracing. Okay, here we actually see our P wave, which is what occurs in atrial depolarization, back when the signal's moving from the from the SA node to the AV node. Here we have our QRS wave, which is our ventricular depolarization as the signal moves down through the ventricles. Here we have our T wave, which is the signal we can read when our ventricles repolarize. And we know that our atria repolarize during the QRS wave. That's not what the QRS wave is showing. The QRS wave is showing the ventricles depolarizing, but that is such a big electrical signal that it masks or covers up the atria repolarizing. So we know the atria do repolarize during this time. Okay, so again, here's our graph of what's going on in our intrinsic conducting system and those nodal or autorhythmic cells. Again, we have our pacemaker potential, also called our pre-potential. Again, that's caused by sodium leak channels and sodium leaking in. Once we reach threshold, which happens to be at negative 40 millivolts, then we actually open up our fast calcium channels. Calcium rushes in. That's our depolarization. We cross zero. That's called reverse membrane potential, which triggers our potassium gates to open up. Potassium gates open up and potassium leaves. So that's our repolarization. And then we reach our resting membrane potential. And then we have our leakage in of our sodium, and we go through the graph over and over and over. And of course, this is always initiated in the SA node, but if something happens to the SA node, the AV node has the ability to take over and to go through this spontaneous depolarization also. Again, this is the graph for an autorhythmic cell where we're sending the electrical impulse, and this is the graph that we would find for the actual cardiac muscle cell, which makes up 99% of the cells in the heart, where the intrinsic conducting system only makes up about 1%. Now, again, here's our, notice we have a different resting membrane potential here than we did in the autorhythmic cell. It's lower, it's actually negative 90. This does not happen spontaneously. We have to have a stimulus to trigger a very quick threshold barely above negative 90. And of course, that stimulus is the fact that we have gap junctions allowing ions to cross from the autorhythmic cells over into the contractile cells. And then contractile cells have gap junctions to contractile cells. So what's going on in one will pass ions to the next cell. Once the ions come across to raise the voltage from our resting membrane potential to this very quick threshold right here, then that's going to open up fast sodium channels, and sodium is going to rush in. Now, these are fast sodium channels. In the above graph, when sodium leaked in, notice it's more gradual. It didn't just shoot straight up. So this is sodium leaking in. This is sodium rushing in. So this stage is our depolarization. Notice rapid depolarization due to sodium entry. Now, once we cross zero, again, we reverse membrane potential. And what that does, that starts shutting these sodium channels, but it actually opens up some calcium channels. So calcium starts coming in, but instead of rushing in, calcium is coming in slowly. So it's actually slow calcium channels. So instead of this climbing up higher and higher as it did when sodium came in, then calcium kind of causes it to level off. In the meantime, we actually get potassium channels starting to open up, and then after a certain amount of time, the calcium channels shut, but the potassium channels continue to open, so we get the last part of our graph, which is our repolarization, which is basically potassium leaving, and that's basically the same thing that happened here. Our repolarization came when potassium left. Now, one thing important about this is that this plateau area, the second portion that we call the plateau area, it's always the same duration and length, which gives us a timing of how long it takes our muscles to contract. Because the whole key to the heart muscles is that we coordinate when they contract and for how long they contract, so we can close the chambers and effectively squeeze blood out of the chambers.